Andreas is going to present developing a standard interface for sets of k-mer sets index structures. So please try to share, so not your entire screen, but the program you use for PDF. Okay, looks fine. Please go ahead.
Yeah, thank you for your introduction. Yeah, as was told, I'm talking about developing a standard interface for sets of k-mer sets index structures. And for those of you who might be unfamiliar with the term sets of k-mer sets, I start with a brief explanation of the term. And actually, this is also my motivation. So we are in the field of computational pangenomics, and there you often have the problem that you have to process thousands of input sequences within a reasonable time and within the limits of main memory. And to construct a compact index that allows fast queries is therefore a hard task. And normally what you have is a set of genomes, for example, that you would like to compare or create the index of. Or what's also possible and also often in practice is that you have a set of RNA-seq experiments in the sequence read archive, for example. So a set of reads that you want to index. And instead of indexing these whole large sequences, a common approach is to use a set of k-mer sets. So each genome or RNA-seq experiment is represented as a set of k-mers. And in total, then you have a set of k-mer sets. So this would be my whole set of k-mer sets. And so why do we want to talk about defining a standard interface? Let's start with the motivation. Actually, let's first talk about possible representations of these k-mer sets. So the first representation you see on the left is for each of the input data sets, so genomes or experiments, it's possible to store the set of k-mers. Or another point of view is for each k-mer is to indicate in which of the input data the k-mer occurs. This is typically referred to as colors. So for example, for this k-mer AACG, we would store that it appears in genome or RNA-seq experiment A, and so on. And this is usually the representation that many of the tools that are available use. So now let's come to a motivation. So why talk about a standard interface? So you see here there are a lot of tools that are already available, and so I will not present you yet another tool to add to that list, but a motivation why we should think about a standard interface for these tools. And the motivation is from a point of view of a user of these tools, and consider the typical biologist, for example, that wants to create an index and query this index. And the typical knowledge of biologists is they maybe know a little bit of Bash or Python programming, and are very happy if they, once their pipeline is running, and they hope that they never need to touch this pipeline again and can use it for all the future. And this is what they hope, but it's often the case that, for example, if a user uses program A for a few years, and after a few years, the development of the program A is not developed anymore, and he has to switch to another program, or program A is developed, but the user finds another tool that fits his demands better. And so what he would like to do is just change every call to program A to program B, and just keep the rest of the pipeline as it is, without having to rewrite the code very much. But as you have seen, there are many tools available, and we will look at some of them in a minute. And you will see that many of them already implement such ideas, so functions to index and query sequences, but the naming of these features will generally differ between them. So let's start, for example, with SecondHello. So SecondHello is a program that allows to index and query RNA-seq experiments. What SecondHello does is it hashes the k-mers using the so-called Otello hashing. With this, it places the k-mers in different buckets first, and then for each bucket, it searches the encoding of the k-mers that allows the most compact representation. This could be, for example, a value list. So for each k-mer, store the position where it appears in the input data. A delta list, so store the position of the first k-mer and then the delta to the next position, so of the next k-mer. Or a bitmap, so store each k-mer as a bit vector and use a 0 or 1 respectively if the k-mer appears or does not appear in the experiment. And then once you have this established, you can just input your sequence queries and then look up efficiently in which of the experiments that you originally have the particular k-mer appears, and then calculate a ratio which of the k-mers appear in the most of the sequences. So which is probably the experiment that the input data, the query data belongs to. So let's look at the implementation side. So how are these features called in SecondHello? Well, first you need to use a k-mer counter jellyfish to count the k-mers in the input data. Then you need to call preprocess group and build to construct your index, and then you have the option to call query to perform a transcripts query. Mantis is another tool. So Mantis also uses a hashing-based approach. So it looks at the different experiments and at the k-mers which are present in these experiments, and it uses a so-called color quotient filter to create a table that maps each k-mer to a certain color ID, and then in the end you obtain a set of bit vectors that tell you does this k-mer appear in experiment 1, 2, 3, and so on, and if yes, it's represented as a 0, as a 1, and if no, it's represented as a 0. So how does the interface look like? So again, you use a k-mer counter to count the k-mers first. This time it's Quaker. And then you call mantis build to construct the index. You have another option, mantis mst, to compress the index. And then again, like SecondHello, you have the option query to perform a sequence query. The next tool is Bixi. So Bixi uses an approach called Bloom filters. So it does not use only one hash function for each k-mer, but 3 hash functions, for example. And for each set of k-mers, it obtains a set of positions in a bit vector that will be set to 1. So in this example, each k-mer has 3 positions in a bit vector that are set to 1. And this would be the resulting bit vector. And the interface looks like here. So it uses MC Cortex, another tool, to first build a graph representation out of the input data. And then you use Bixi Bloom and Bixi Build to create the Bloom filters and then construct the index. And now to perform a sequence query, you call Bixi Search. COPS uses a similar approach, so it again uses a set of hash functions that it applies to the k-mers. Additionally, it extracts the rows in the hash table in the resulting hash table that are relevant and combines them. So only rows, so only the columns in which where every bit is set to 1 for each k-mer will be relevant. So if, for example, a k-mer appears in the experimental data 0, D0 and D1, then you will obtain in the end a bit vector consisting of 1, 1 for D0 and D1 and 0 for all other experiments. And now if you have obtained that, you just combine the rows by adding them, and you get a measure which of the experiments is likely the one the set of k-mers that your query contains belongs to. And again, the interface. So there's the option compact construct to construct the index, and the option query to perform a sequence query. Then another approach that is shared by multiple tools are sequence bloom trees. And here the idea is, again, you have bloom filters that you apply to your k-mers and obtain bit vectors, but now they are organized in a tree structure so that every child node, every parent node is basically a union of the bit vectors of the child nodes, as you see here, for example. So this parent here is a union of these two bit vectors, so every bit that was set to 1 in at least one of the child nodes is set to 1 in the parent node. And for example, SPT and SSPT, these were the first tools implementing this approach. And there the interface looks as you see here. So you first call hashes, count and build to construct the index. You could can call then compress to compress the index, and at last you call query to perform a sequence query. How DSBT is another tool that makes use of sequence bloom trees, which was a slight modification that I won't go into detail now, but there the interface looks as follows: make PF, cluster and build to construct the index, and query to perform a sequence query. Now let's come to graph-based approaches. So we've already seen in the previous talks that the data structure called the Brine graphs, which you can use to nicely represent k-mers as a graph and neighboring k-mers connected by edges. And the first tool that I would like to present you is Bifrost, which first can construct an uncompacted de Bruijn graph, as you have seen, but also to save a bit of memory, can compact this de Bruijn graph. So as we've seen in the previous talk, merge non-branched paths like this one to actually store this path in a single node in the graph. And there you have the options Bifrost build to construct the index, Bifrost update to add other sequences to the index, so to update the index after you have constructed it once, and Bifrost query to perform sequence queries. And with these graph-based approaches, let's first see another example. Vari or Cosmo, it also implements the de Bruijn graph, but another kind of compaction, which is called succinct de Bruijn graph. And the interface is Cosmo build to construct the index, pack color to compress the index, and variant merge, vary merge or color merge to merge graphs and update the index. And as you've already seen here, with the graph-based approaches, there are a few more possibilities compared to only constructing and querying the index. And for example, Cortex, another tool, allows to actually inspect the graph and find interesting regions. For example, if you have different genomes that you represent in the same graphs, you could look for heterozygous or homozygous regions. for repeats or for polymorphic sites. And so with that, there are some more options that go beyond to basically constructing the index. So to construct the index, there are first the options SE-list, PE-list for single and paired entries, and color-list to construct a colored de Bruijn graph then. And now, as I said, there are more options, for example, detect bubbles in the graph and output the bubbles, and use that for variant calling, for example. And a similar tool is MC-Cortex, which also allows to build the index with the options build and index, and then, for example, clean and correct errors in the graph that come from errors in the reads, or output unit ticks or assemble context even. Now let's come back to our motivation. As you have probably seen now, since we have looked at the tools, what they actually implement, it's not that easy as we have wished. So, for example, only the indexing command, in some tools it's called index, in some tools it's called build. And so for the user or even for the programmer that wants to use these tools using their application programming interface, it's often hard to adapt to another tool. It's not as simple as just switching the name of the tools in general. And that is actually the aim of my programs. So if we can define a common standard interface, so a set of features that all tools should basically implement, and a consistent naming that these tools have, we could actually make it much easier for the users and programmers that are working with these tools to switch between tools, to see what other tools like and are like, and to fit the tools that fits their demands best. And for that, I have three steps in this project that I want to pursue. So first of all, contact the authors of the existing tools that you've seen before, and then with them together decide on a set of core features and consistent naming. And then either the authors would agree to update their tools with the interface that was defined, or we could write a wrapper that is implementing the interface and internally calls the features of the tool as it was previously called. And I'm very happy that some authors of the tools that I showed you before already responded to my email. So it will soon be put into motion. The second step then, first of all, let's look at a possible index interface as we could establish it. So first, I separate it into a core interface and an additional interface. The core interface could be features that all tools should more or less be able to perform, so should implement. And this is, as we've seen in most tools, to construct the index, and this can be done by inputting a set of sequences, so assembled genomes, read sets, or set of k-mers, each with a distinct color, so a different set of input files, basically, and the output would be an index. Another feature would be to add or remove sequences to the index after it has been established. So you get a set of sequences in an existing index and output the updated index. One feature could be to read or write the index to a file, so in proprietary format, in GFA format, FASTA format, or basically a k-mer matrix. Then you could query for sequences. So for k-mers, for example, you input a k-mer and obtain a set of colors in which this k-mer is present. Or in the other direction, you could input a color vector and output all k-mers that are present in this experiment, for example, or a genome. So this would be the core interface. An optional interface, for example, that not all tools could implement, or some tools could implement better based on their internal implementation. Would be a general query, for example, so a set of sequences or a set of colors, or any quorum, so a minimum ratio of colors that should be matched, and output would be all hits that are matching the query. Forward backward extension, this would be especially for the graph-based approaches, so you can input a k-mer or unitic and find the neighboring k-mers or unitics in the boron graph. And last but not least, a abundancy query. Find k-mers or longer sequences. You input k-mers or longer sequences, for example, transcripts and the color, and outputs abundance. So not only the presence or absence of the k-mer, but the abundance in the sequence in the color. And there can, of course, be additional tools that are use case specific, for example, genome assembly tools, as we've seen in, for example, MC Cortex or Cortex, to filter erroneous k-mers to assemble context. Phylogenomics, as we've seen in the tool, in the talk of Roland Bittler, for example, so output a list of splits for a k-mer or color set, and you can think of many more. So this would be the first step to get into contact with the authors and to decide on a core interface and optional and additional interfaces. The second step that I would like to perform is set up an automated test environment. So I would also like to evaluate the existing tools. And for that, I would like to, again, with the authors together, decide on a suitable test data set and criteria for the evaluation, for example, the runtime, the memory usage of the tool, but also the false positive rate, for example, which is especially relevant for tools that are using Bloom filters. And then I would like to implement a benchmarking platform where every tool could be evaluated in the public, probably in the DenB cloud, so the cloud service of the German network for bioinformatics. And the last step would be to perform the actual study and to publish the results. So in the end, we could, for example, provide an open catalog with the results. So for each tool, put a description of the tool, put a link to the GitHub page, and show the results. So does it implement the core interface, the optional interface, and which of the additional use case specific interfaces does it implement? The results of the benchmarking study, so for example, the runtime and memory usage, how it performs compared to other tools. And with that, for example, any user or programmer that is interested in using one of the tools could just browse through the catalog and find the tool that fits his demands best. And with that, I want to conclude my talk. So we've seen that pangenomes can hold a variety of informations that there are many tools that already implement pangenome-based, k-mer-based index structures, but no consistent interface on the functions and the naming of the functions. But with the aim of the project, establishing a standard interface, we could enrich the comparative genomics and genome research as a whole. I want to thank my supervisor Jens Steuer and the whole genome informatics group in Bielefeld, my graduate school, Digital Infrastructure for the Life Sciences. I've put here my email for contact in case any one of you who has a tool who was not listed yet or who I did not contact yet wants to participate in this benchmarking study and in the definition of the interface. So you can at any time contact me. And thank you for your attention.
Thank you very much. Nice talk. And we can go to the questions. The first one is if there is any reason why Mantis was not in the final list of tools.
You mean in the one of the last slides, the list? This was the list of tools where I already contacted the author, and the author already responded to my email. So Mantis, I did not yet contact the author, so it could be part of our list in the future once I get a feedback from the author.
Okay, that was a question from YouTube, from the YouTube chat. Mm-hmm. The second question is, do you think it would be possible to aid the user in choosing the best tool to use according to the characteristics of the inputs?
Yeah, I think actually with the example of the catalog that I showed, we could also implement, for example, a search, and the user could search which are his demands. For example, if he's interested in genome assembly, he could filter for the tools that support this interface, or if runtime is the limiting factor, or if memory is the limiting factor, he could sort the tools, for example, by the lowest memory or runtime. And we could actually, I think, aid the user in finding the tool that fits its demands best. Yeah.
Okay. Now, are there any more questions? It looks like there are no more questions, so thank you